So we are 3 minutes over 10 a.m. So maybe we are good to start. Well, good morning, everybody, or good afternoon, or good night, wherever you are, and happy St. Patrick's Day for everybody. So welcome to Speaking of Oats. First, I will introduce myself. My name is Pablo Olivera. I am a faculty at the Department of Plant Pathology at the University of Minnesota. And since the last week, I am the new director of Oat Global, taking over the leadership from founding director Jim Bradin. And I want to take this opportunity to sincerely thank Jim for all his contribution to the oat community during the last 6 years and wishing him the best on the new stage of his career. So speaking of oats, it's a partnership between the Oat Newsletter and Oat Global. Charlene White, editor of the Oat Newsletter, is with us today and will be handling the question and answers after the seminar. Oat Global is a public-private partnership that aims to coordinate and connect all researchers and oat extension professionals worldwide with each other and with individuals along the entire oat value chain. We are glad you are here today. But before I introduce today's speaker, I want to take a moment to remind everyone that you can use the Q&A widget to ask questions throughout the presentation. But please use the Q&A widget and not the chat function for questions, since these preserve the question for later access. Today's session is being recorded and will be made available later via YouTube. So today's speaker is Dr. Weikai Yan, a research scientist and oat breeder at the Ottawa Research and Development Center of Agriculture and Agri-Food, Canada. So I went through Weikai profile and I was highly impressed on his research and publication record, including a couple of books on biplot analysis and crop variety trials. Today, he will talk about mega-environment analysis and breeding for mega-environment specific oat cultivars for Canada. Weikai, thank you for being here today, and the floor is yours. So you can share your screen. And you should unmute yourself. Weikai, you are mute.
Sorry about that. Hello, everyone. And first of all, I want to thank Charlene for inviting me for the talk. And I want to thank Pablo and Annie, the Oat Global Center, for setting up everything. And as you see, my topic is mega-environment analysis and breeding for mega-environment analysis, specific oat cultivars for Canada. So why are we doing mega-environment analysis? Our purpose is to exploit specific adaptation to improve selection reliability and to enhance breeding progress and to increase regional and therefore global production. So mega-environments is a big word, but they are actually subregions of a crop species growing regions. We all are familiar with subregions. Subregions can be defined by the latitude, the longitude. For example, in Canada, we often have Eastern Canada versus Western Canada. And for individual provinces, for example, in Quebec, the agricultural region was divided into 3 zones based on the flowering time of 3 plant species. In Ontario, the crop regions were divided into 6 heat unit areas. And in the West, in Canadian West, was divided by soil zones. We got 3 soil zones and plus irrigation area. Those subregions are still widely used and very useful, but they may not be accurate enough to guide breeding and cultivar recommendation. So by mega-environments here, we mean subregions based on data from multi-environmental trials. The criteria to divide into divide the green region into subregions. It's a which one where pattern. There are some, I am quoting here from Gonzalo. He said, if a winning cultivar is different in different environments, then the region should be divided into mega environments. The basis to divide subregions is GGE, which G means genotypic main effects, and GE means genotype by environment interaction. Those are the two items that are relevant and must be considered when we are doing genotype evaluation, environment evaluation, and mega environment analysis. Here, GGE means G plus GE, or G over GE, or G versus GE. The GGE term appeared to be come from myself in 2000, but the idea has been there 85 years ago. All major publications, analyses have the idea of a GGE in them. In terms of GGE or which one where patterns, we have three different ways to present them. The very famous one, and everybody knows, it's the joint regression chart. In this chart, a cultivar is displayed as a linear line. It's a linear regression against the environmental means. So each line here represents a cultivar. Now, if two lines are parallel, there's no G by E between them. If two lines are not parallel but not crossover, then the G by E is not big enough to cause rank changes. So those are not relevant for selection. But if the G by E is large enough to make two lines cross, this is called crossover G by E. You see, we got lots of crossovers here, but not all crossovers are equally important. Only this black line and this red line make sense because G1, G2 crosses, which divides. That means that G1 was the highest yielder in this area, and G2 was highest yielder in mega environment 2. Therefore, the mega environments are defined by the winning cultivars. And here also it's by the productivity level of the environments. So this is this chart started in 1938 and carried over even today. It's a conceptually very useful chart for understanding G by E. So this is how people dividing a green region into subregions. Then come to another chart, which is supposed to be better, but similar. The only difference is the x-axis. Here, instead of being environmental means, we have the interrogative PC1 scores of the environments. Now, again, you see the crossovers and the more meaningful crossovers, G1 and G2 and G3. Those define mega environment 1, mega environment 2, and mega environment 3, because G1 was the highest yielder in ME1, G2 the highest yielder in ME2, and so on. So the x-axis is interactive scores. So this is different from the previous one. Now come to the third chart with the GG biplot. This GG biplot is based on two principal components here. The the blue letters is presenting cultivars, and the red letters represent the environments. Now, first of all, we use a polygon to contain all the cultivars. Then we draw radiative lines from the biplot origin to perpendicular to each side of the polygon. So those radiated lines divided the biplot into regions. In this region, we got 7 environments, and the highest yielding cultivar is friendly. And in this region, we have 2 environments, and the highest yielding cultivar is Zavid. So this is how we see which one where. Compare those 3 types of charts. The GG biplot is more efficient because it explains more G by E and genotype, and G and G by E. It's more easy to construct and allows more entries to be displayed. It has other functions, so it's more versatile, and some say it's more elegant. There's another important point though. So mega environments must be defined by repeatable patterns, not just a pattern 1 year. So how do we do that? It's a very challenging topic. And some say, well, you just have to look at the GG biplot each year and then look at if the patterns are similar. This is the easiest said, but difficult to say. Here's an example. This is a GG biplot for the oat yield trials in Quebec in 2006. Here, this is another form of biplot, which shows the genetic correlation between locations. If the angles are small, then they are positively correlated. If the angles are greater than 90 degrees, then they are negatively associated. And it's really defined by the relative magnitude of a genotype main effect and G by interaction. Now, if we want to see the repeatability, we have to make one plot for each year. And we did see some similarities across the years, but this is not easy to see. So recently, I figured out a way to put all those different charts in a single biplot. It's like this. This biplot is called a location grouping biplot. It combines all 6 charts together. Well, if this still does not make sense, let's convert this to a different view. This chart, each location is represented by all the trials as a location like this. Now, if the 2 locations are correlated all the time, then they belong to the same mega environment. If 2 locations are always different, then they belong to different mega environments. If 2 locations sometimes closely related, sometimes not, they are still belong to the same mega environment, except that the mega environment is more variable. So this chart divides those locations into 2 groups. The first group contains zone 1 of Quebec plus Ottawa plus another location from zone 3. The other group, however, are from zone 3 and zone 4 of Quebec. And you can see within each group, the location cannot be separated, but between the groups, they are separated pretty well. Therefore, we see that the Quebec oat growing region consists of 2 mega environments. two mega environments. They are more or less separated. But within each mega environment, there's still a lot of variation. And those variations are called unrepeatable G by E or genotype by environment interaction. Of course, the mega environment will be reflected by which one where. For example, in the first mega environment, orange was the highest yielding cultivar, but for the second, Bia was the highest yield cultivar. This is for the 206, 210 data. And later we followed up the data until last year, the pattern still there. Using the same strategy, we analyzed the locations in Eastern Canada. In the Eastern Canada, including Ontario, Quebec, and the maritime provinces. And we found two contrasting mega environments. The first one was the area 2 and 3 of Ontario. And this mega environment is characterized by very heavy crown rust pressures. And the second mega environment is the rest of Eastern Canada, including Northern Ontario, Quebec, and the Maritimes. And of course, the different mega environments have different cultivars. That's the point, right? And use the same strategy, we analyzed trials across Canada. We found that the Canadian oat green regions can be divided into 3 mega environments. Mega environment 1, Southern Ontario. Mega environment 2, the other regions of Eastern Canada. And mega environment 3, all the Canadian prairies, including Manitoba, Alberta, and Saskatchewan. So now we see that the mega environments are not the same as the agro-ecological zones that we are familiar with. For example, in Quebec, some locations classified as zone 3, but actually it behaves like a zone 1 location. In Ontario, the six temperature zones were classified into 2 mega environments. And the prairies or soil zones belong to a single mega environment. So why do we want to do a mega environment analysis? One reason is to improve the heritability. And heritability, it's also called reliability of selection. This equation describes how heritability is calculated. Look at this part. The variance for genotype by environment interaction reduces heritability. Now, if we select across all the regions, we are dealing with all the G by interactions together. However, if we are selecting within mega environment, the G by E was reduced by half. Therefore, the heritability will be higher. The selection will be more reliable. This is one major part of major step to deal with G by E and to improve heritability. Another part is to increase the number of locations and years. Because you got more locations and more years, and this part will be smaller, and therefore heritability will be improved. And now everybody is talking about genomic selection, right? So genomic selection can be understood the same way. First of all, we need to develop genomic selection models for each mega environment so that this genotype by model variance is reduced. Secondly, we need more models. That means more training data from more locations and years. If you compare the conventional heritability versus the heritability for genomic selection, one major advantage of genomic selection is that this M or number of years can be increased indefinitely. So therefore, at some time, this genotype by model will disappear and the heritability will approach 1. Therefore, anything you produce good will be really good. Now time to talk about how to breed in mega environment specific cultivars for Canada. First of all, we need to understand the mega environments as we did. Secondly, we need to have some crosses and populations that have the potential to select for different mega environments. Thirdly, we need to do some exploratory trials that are covering all mega environments so that we got an idea which breeding line is likely adapted to which mega environment. Then we put that line into that specific mega environment for further test. So here's a real story. So we made some crosses in 2013. And in 2018, we came up with 66 lines that were tested across Canada. From those, we selected 17 lines and tested it again across Canada. When we put those 2 years data together in a biplot, we saw here one line, OA 1627-1, was particularly good for mega environment 3, which is Western Canada, but it was not good in the other mega environments. So if we look at this data just for mega environment 1, mega environment 2, we saw this line was the best in mega environment 2, and that line was best in mega environment 1. So based on this pattern, we submitted a 1627-1 for the prairies. And when we continue to test the other 2 in the Ottawa Research Center trials. After 2 years, we got those results. In Western Canada, OA 1627-1 indeed yielded very well. It actually yielded better than all the checks. So it was supported for registration this year for the West. And then the other 2 lines, OA 1644-13 was the best for mega environment 1. But it was pretty bad in the mega environment 2. And in mega environment 2, OA 1623-5 was the best, and which was not the best in mega environment 1. So you can see the mega environment specific adaptations. Looks like the 2 years nationwide test was sufficient to identify the highest yielding cultivars for each mega environment. This is, and then why do we want to continue to test for more years? Because the data are required for the recommendation committees to support. So let's summarize what I have said. Number 1, mega environments have to be defined by repeatable GGE patterns, not just GE patterns, as you see. And the location group biplot was an effective tool for mega environment analysis. And to handle genotype by environment interaction, And we need to go 3 steps. First, to divide the target region into mega environments. Second, to select within mega environments. And thirdly, to test adequately. That means you have to have enough location in years. And in our case, actually, we don't really need a lot of locations or years. Maybe 3 locations per mega environment, and 2 years will be good enough to identify the best cultivars for each mega environment. Now, how to breed mega environment specific cultivars? We presented a true story, which has 3 steps. First of all, you make some hopeful populations, breeding populations. Second, you do a preliminary screening cross mega environments. And third, put the specific, and we put the specifically adapted lines into specific trials. That's all what we did. Coming in the future, I was thinking maybe the logical ways to make some mega environment specific crosses and testing only within mega environments. There are some pros and cons to do this, but logically, that's what we should do. Or we should use mega environment specific genomic selection models to replace the preliminary screening. So that we will not have to test nationwide. We still got some ideas and where to test what. So basically, that's what I have to present. And if you don't understand well, I apologize for my English. So acknowledgement. First of all, the 2 technicians of my group, Brad, the wrong spelling. Brad Dehan and Matt Hayes. Those guys, without those guys, I don't know. I probably don't really have much to do. I probably cannot do much. And the collaborators conducting the trials. Many people, I don't even have a space to list them. And the old quality team, which is always important for us to breed good milling oat. And the disease team in this program. And the breeders, the old breeders in Canada have a very good tradition to work together. So they are very important. Here I have Mike Mullen from North Dakota. Mike was one of the earth people to get the anchor together. And finally, I have to thank Oat Global and Charlene. Now I have a dream breeder here. Actually, Pablo replaced him and Annie. Thank you so much.
Thank you, Weikai. That was really great. Yeah, there's an awful lot in there. I think the mega environment thing is a really, really interesting one, the whole topic. And actually, in fact, I'm kind of overwhelmed by all the information you presented, and it's just so cool. One of the things actually I would like to say first, though, before we get into discussing that, I would like to welcome Pablo to the community. As you mentioned, Jim isn't director of Global anymore. We now have Pablo. So just wanted to give you a welcome to the community. And you have some really big shoes to fill because, you know, Jim did an awful lot of wonderful things, including setting up this webinar series. So, so we look forward to great things from Pablo. And Weikai, as I said, of course, you've done some really amazing things too over the years. The GGE biplot thing, for example, all your books and everything else, as Pablo mentioned. And I wanted to let people know, so you're working actually on a big article right now, right? Concerning mega environments for crop science, is that right?
Yes.
And so when are you expecting that might be published?
I think I got the deadline for July of somewhere.
Okay, so it'll be sometime after July, then that will get published. Yeah, right. So that's something we have to look forward to. So we have a question here from Tony Yang. Thank you for the presentation. It is very impressive. Just wondering if it is possible to share the PowerPoint with us later by emails, and it's hard to write down all of the key information during the presentation. And he says, thank you. I will mention to that myself that this seminar is being recorded. So in terms of that, people will be able to go back and actually hear Weikai speak about this all again. I suppose some people might want to analyze the slides a little bit more. So Weikai, you can answer.
No, I'm always for it. Anyone wants a copy, they can ask from me or from you.
Okay, thank you.
Just so people know, every Speaking of Oats webinar goes on YouTube within a few days after. We have not done this traditionally, but it is possible to post the slide decks to either the Oat Global website or email it out at a later date. So yeah, flexible on that.
Right. And the other thing I do, of course, I post the links in the Oat newsletter, and I've actually also been putting the links in our research reports sections on that page. And if you want, I could also post the PowerPoints there, actually.
No, I have no problem to share, Charlene. So, anyone wants a copy, go ahead. So ask me or you,
we'll get it out. So great, thank you. So more questions. Catherine Howarth says, if you're looking at milling quality traits, do you see the same effect of mega environments?
No. Actually, the milling quality traits, including growth percentage, beta-glucan, those things, they have a pretty high heritability. That means you can, if you see a good miller in Quebec, it's probably also good miller in Western Canada or in Ontario. So there's not much, there's always G by E, but not much G by E to divide into mega environments. So when we talk about mega environments, we are mainly talking about the yield.
Right, that makes sense. So anything with high heritability, then it's not going
to right. Well, high heritability means that very little G by E, right? Very little G by E means that the whole world is a single mega environment.
Well, okay, and actually something came to my mind too, sort of generally speaking. So you were sort of overlaying these mega environments with the more sort of traditional ways of breaking up and, breaking things into environments. And, it's kind of counterintuitive to say that, well, agroclimatic zones don't match up to this mega environment pattern and so on and so forth. So what do you think is actually underlying what defines each of these mega environments in Canada? I mean, if it's not the soils type or the climate, this type of thing, do you have any ideas of what it is?
Well, actually, you are asking the genetic basis of G by E or environmental basis of G by E. Of course, when we talk about the genotype by environment interaction, we are not just talking about the environments or the genotypes. We talk about the interactions. If there's a gene respond differently to a different environment, you got G by E, right? But if there's no such different responses, then you don't get G by E. Yeah, but normally we would say that the major genes always got some G by E. For example, genes controlling large gene resistance, gene controlling maturity. And if probably we don't have a winter wheat people in this group, but if you look at the winter wheat, you got genes for photoperiod responses, genes for vernalization requirements, those major big genes always are important reasons for responding differently to different environmental factors. And also these major genes, no major gene, not a single gene has a single effect. All the major genes have a multiple effects. Bad effects or good effects. That's why breeding is so difficult and so interesting.
Yeah, that's very true. Hey, there's also a question here from Juan Arbalez. Dr. Yan, thanks a lot. As you define these mega environments, you begin to create specific populations or crosses for those specific environments and testing strategies for each mega environment, etc. Do you have any advice on how to allocate resources across each mega environment? How many crosses for each environment, etc.? What variables should you consider to allocate these resources?
That's a very big question. If you look at the big companies, the big companies always focus on the mega environments that are big in terms of acreage. That way, they can select a single cultivar to grow a lot of acreage and to be more profitable. But for non-profit companies or institutions like us, Southern Ontario is a very small acreage, and about 30,000 hectares, but it's a very complicated mega environment. So it's not easy to breed good cultivars for Southern Ontario. So basically the number of locations required should be proportional to the complexity of the G/E in the mega environment. So if you have a mega environment that has very little G/E, then 1 or 2 locations would be good enough. But if you have a mega environment that is very complicated and the year difference is so big you cannot predict, then you need more locations and more years. Number of crosses, and it's all depending on how well your crosses are. If you have a good cross, 1 be good enough. And sometimes you make 100 crosses and don't have a good one to select a good cultivar. But from my experience, you will always have a good cultivar as your parent, and to put some new genes or possibly new genes into the adapted cultivar, so that you increase your chance to get good cultivars from it. I wonder if I answer your questions.
Well, I guess Juan can let us know if that answered his question. I suspect it did. We'll move on to Nick Tinker's question. Great talk, Weikai, very clear. My question is related to Charlene's. How well can we count on ME based on test sites to represent ME of producers? Or is there a danger that some part of these patterns are based on the uniqueness of cooperator test sites, planting time, weed control, soil, harvest method, etc.?
That's really a good question. Usually the locations are selected for convenience. And there are areas probably are not well represented by those locations because not many farmers are willing to do your test, and not many farmers have the capacity to do the test. But that's why we are talking about multiple years and multiple locations, right? Because if you do multiple years, the locations that are not well presented in 1 year might be represented in another year. So that's how we do the selections.
Yeah, and I guess these days too, I mean, we've seen it here at Ottawa. You can have quite different extremes during different years, you know, from cold and really, really wet to sort of such bad drought. Plants are only maybe 6 inches high. Yeah, so you're still going to need the multiple years and that sort of thing.
Yeah, probably more years in the future because we are seeing more severe and frequent extreme weathers in recent years. Last year, Western Canada suffered a lot from the drought, and there are many crop failures, and some breeders don't even have enough seeds for testing. Mm-hmm. So this large fluctuation, extreme weathers. When you look at the average is still there, right? But when you look at individual years, are really extreme. And last year we actually lost the trial in Quebec because of frost. We never had that before. So looks like we all really have to increase our diversity in our breeding population, and we'll have to test at more locations and more years, and to buffer that kind of extreme weather.
Right, yeah, because it does seem, at least locally, to be getting more extreme.
Yeah, so those, in terms of G/E analysis, and those are the unrepeatable G/Es, and there's nothing you can do except to add more locations and years.
Right, yeah. I guess too, you know, originally I was really surprised to find out that Western Canada was just that one big mega environment. And I think that was partly because I know that in, say, Manitoba, you know, versus Saskatchewan, I mean, the prevalence of rust in Manitoba, you know, that's such a big factor, and it's much wetter and so on and so forth, generally speaking. And I guess, though, I mean, rust genes are major genes, the simple ones are anyway. And so I guess you have to keep in mind, though, not only breeding for your mega environment, but then you also have to make sure that for particular locations, you would be incorporating something, say, like the rust resistance genes and so on, right? So do you see that as a couple of different layers of like when you're thinking of this as a breeder breeding for, say, the West, for example, you're still going to need rust resistance in that potential cultivar, right?
Right.
So
yeah, so because rust in the West probably are not like a rust in Southern Ontario, which is always there and really dominant. And in the past few years, we've been seeing data that in the Ontario Cereal Crops Committee trials, if you got the PCG in there, you got 25% higher yield. If you don't have it, you lose 25% yield. Right. But once it's sprayed, then that difference disappeared.
Yeah.
But in the West, probably the rust is not happening every year. Therefore, it's considered as unrepresentative or non-repeatable G/E.
And this is why we still need breeders.
Right.
Okay, so we do have some other questions here. So Juan is saying again, cooperative trials are sometimes used to define mega environments. Some have 24 unique entries and others have 36 entries, etc. Do you have any comments on the size and composition of the populations used to accurately define these mega environments?
Yeah, well, the general comment would be that the more entries you have, the more results probably will be reliable. But this, I'll talk about the location grouping method. What's good about this method is that it does not require does not require the same cultivars appear every year. And really does not require any common cultivars. So if you got a trial, similar locations, and you are okay to use the method to look at. Quite a few number publications in crop science in the last 2 years using this method. So the algae biplot is pretty flexible. in terms of a number of locations and the number of entries each year.
Okay. Thanks. And Bruce has a question too. Should we be spending more effort on drought tolerance in oats as seen in efforts for corn, soy, and wheat in the US? Should drought tolerance work across all mega environments?
Hi, Bruce. And nice to hear from you.
By the way, yeah.
Yeah, I think that's a very good idea. That's an important idea. Really, we have seen the drought is never this bad before, and it's more severe and more frequent. You know, breeders, the breeders not a physiologist, so they don't do physiological studies or screening at the controlled environments. But if this trend continues, probably we should, you know, set up some artificial screening nurseries for drought, just like we did for corn rust or some other major stresses. And that will particularly will be important to screening for drought tolerant germplasm that can be used for making crosses. But for cultivar release perspective, we also just have to test at more locations and more years. And this is our real-time method to deal with climate change and whatever stresses in there.
Yeah, that's true. Thanks. Catherine has another question. Do you think that grain yields in oats are much more variable between trials in a given mega environment than in other cereals such as wheat or barley?
I don't really know, Catherine. I have not compared oat with other crops, but also I have to say that when we say mega environments, we're talking about specific to a crop, right? That's why I'm talking about oat mega environments. Oat mega environments may not be the same as corn mega environments or winter wheat mega environments. So I think each crop should have its own mega environment based on experimental data. That'll be more accurate. And I think the corn heat unit in Ontario are still working very well for corn and soybeans, but not for oats or barley or spring wheat.
Yeah, I was kind of wondering the same thing, whether the mega environments would overlap in a similar way for some of the other crops.
Different crops may be similar, but it will not be good to assume they are exactly the same.
Right, yeah. Okay, that's all the questions that are there for the moment. If anyone else has any other questions, please ask them now. Yeah, one of the things, you know, Weikai, I talk to you all the time because, of course, you're at the same center as me. I've known you forever, and you're my brother from another mother, sort of thing, right? So we discuss all these things, and I mean, you're completely brilliant with all of this. And we've had some discussions recently just about the breeder's role in general, and using all these different new techniques. I mean, and one of the things though that always gets me about you is you're so brilliant with all the mathematics and all the, defining such things as these mega environments, et cetera, et cetera. But at the same time, you're very much an artist, and you very much believe in the concept of the breeder's eye. And so I'm wondering if you can just maybe mention that a little bit for a moment, and how you kind of reconcile those two parts of yourself.
Well, I think the breeders in the past, the people are saying that the breeding are 70% of arts and 30% knowledge, but recently that kind of assessment was criticized by some people, but I think the genomic selection looks like will soon replace the breeder's eye, so I'll have no pride on that.
Oh, yeah, I'm not sure that anything will ever truly replace the breeder's eye. But maybe.
But when I'm in the field and when I see a plant or see a plot, I often envision what it would look like when it grows in a bigger field, or in a bigger field in Quebec, a bigger field in Ontario, what it would look like. And always that kind of envisions and anticipations. I don't know, good or bad, that's what often happens to me while I'm in the field.
Because there's so many sort of large scale things going on now. I mean, yes, we have the genomic selection we've been working with, of course. Also, though, there's a lot happening in the area of large scale phenomics and all these types of things, you know, metabolomics and all these other things. And I mean, yes, there will be very, very good for screening large quantities of germplasm and all that kind of thing. But yeah, I mean, I still think that we are definitely going to need people who can envision. So those things would be useful, I think, for screening out a lot of material. We had this conversation with Brian Rossnagel recently about, you know, how the breeder's job is to get rid of stuff and not much else really. But yeah, I think it's going to be very interesting in the future, and quite a challenge for breeders too, because you can collect so much information now on so many different things, and how do you pull all of that together and yet still, as you say, get out in the field and really get to know your material, know what you're working with, when to collect your data, all that sort of stuff too.
Well, yeah, I totally agree with Brian Rossnagel that breeder selection are really culling bad ones, discarding bad ones, not selecting good ones, because it's so easy to eliminate the bad ones. You know, if this line is too late, bad; too tall, bad; too weak, bad; kernel small, bad. There are so many things to discard. So even it's not accurate enough, but when you are looking at 10 traits, 20 traits, you can effectively remove 90% bad ones pretty safely. And genomic selection is taking a total different approach. Genomic selection to select the good ones, and that will be more challenge. No breeder would try to do that in early generations, right? But genomic selection does have the potential to do that theoretically.
Right.
But of course, genomic selection can also take the breeder's approach to discard the bad ones, but then you will have to have 20 models for 20 traits, right? So, that would be a challenge too.
Yeah, a lot of challenges now.
Yeah.
Good ones though. I mean, I think, you know, it's
right.
a very interesting time for plant breeding right now.
Yeah, but then breeders don't have to go to the field, and they have nowhere to go there.
Oh, that's your favorite part. Actually, Whoopi has made a comment here. Weikai is a walking out germplasm database. Whoopi, you're bang on. Weikai really is a walking out germplasm database. I guess we're coming to the end of our time here, and Catherine has put up a reminder that yes, this fall is the International Oat Conference in Australia. Well, fall for the Northern Hemisphere. Of course, it'll, you know, be spring in the South. So registration for the IOC this fall is open, and yes, abstracts are due. Now, actually, I found out the other day, and I will put all of this in the Oat newsletter too, that the abstract deadline has been extended. So actually, you have until May 15th to put together abstracts for the IOC conference. And you can make presentations, as Catherine goes on to say here, in person or virtually. In-person presentations are preferred. And of course, yeah, I mean, I'm really hoping that a lot of people, including me, hopefully we'll be able to get together in person this fall in Australia. And yeah, Weikai, I'm hoping you're going to put together something for the conference.
I've been told to do that.
Oh, I don't know. So at this point, I mean, usually I ask Pablo if he has anything to add to what's been going on, and then we can talk about next month. Do you have anything to add, Pablo?
I think we covered really well. I would like to thank Weikai again. It was such a great presentation, and like to thank everybody to attend the seminar, and also for the good questions that follow up the seminar and the good discussion that came out after. And now I will say that everybody will hear from us about the next seminar. We're still discussing and see when and who's going to give the presentation, but soon they will be hearing from us about it. Yeah.
Right. So as of right now, we don't actually have a speaker for April. We're planning on shifting the schedule a little bit, so more of a every 6 week schedule rather than monthly. But yes, if anyone does want to speak in April, please let one of us know. There's still a couple of possibilities waiting for people to get back to me. But yeah, we'd like to keep this going, of course, and as long as the community is interested in it. So yeah, please let us know if you'd like to speak. I mean, don't be shy. There are all kinds of topics we haven't touched at all in this series. So yeah, anyone's welcome to get in touch and speak, whatever you do in the oat community. Yeah,
I agree. This series has been a great way to keep the community connected, and that's the way we want to keep it going. So even to integrate more people. And so yeah, as Charlene say, if somebody's interested in presenting the research or other topics, please reach her or myself, and we can accommodate a time for your presentation.
And I'm just putting in the chat the actually, I'll put the link to the Oat newsletter to look up the conference information, just because that's what I remember better off my top of my head. .org. So oatnews.org is the site. There's the link in the chat. And from there, you can, if you go under meetings, International Oat Conference, you'll be able to find links to the IOC information there. I believe the website is internationaloat.com, but don't quote me on that. You can check that. I don't have it to hand. So anyway, yes, thank you. And thanks.
Thank you so much, Weikai, again. And thank you, everybody, for attending to the seminar.
Thank you all.
Thanks. Bye, everybody.
Bye.
Bye-bye.